Over to you, Doug.
So thanks very much to the organisers for asking me to speak. Now, big data is a big topic too. And in 20 minutes, I can't realistically do all of it, so I'm going to try and show the overview of the stuff that would count as big data relevant to metabolomics. If you were to go to the literature on big data, you'll often see something called the 3 V's, which you can read there as well as I can, because what makes big data big in some sense isn't always the first one, the volume. Sometimes it can be the speed, less relevant metabolomics. The variety can be, but less relevant. Definitely we want to know the veracity, because how you deal with data depends the extent to which they're actually true. And of course, the main one is the value of the data in the first place. So if you think of a standard metabolomics kind of experiment, a case control study looking for biomarkers in which you've got patients with some disease that's been accurately diagnosed and some controls who you think do not have it, you can collect some metabolomics data, you do a bit of number crunching to find things that are present or absent in one and different in the other, and then you say your job's done, and that's it. And if that were all it wouldn't be a very long talk. But actually, it's somewhat more complex than that. But to make it easier, the canonical variant of that essentially recognizes that many data structures, which is a computer science term, can actually be represented in the form of a matrix or a table, in which typically we recognize the samples or the objects or the things that you're analyzing going down in the rows, and the properties that they possess, for our cases, metabolite levels, going across as the columns, and there will be other columns that you need to remember to take into account, such as the day you turn the mass spectrometer on, and other properties such as gender and age and the like. And we can flip those such that we can seek to use the one and cause a subset of the variables, in other words, the metabolomic data, to be the inputs, and another subset, the thing we're interested in, to be the outputs. And the game then becomes to look through these sets of paired data to determine, for instance, what might be metabolites that are discriminatory as biomarkers. And when you have the inputs and outputs put in that way, we usually think in terms of so-called supervised methods in which we know the class membership, in this case the disease or control, for at least a subset of the data involved, and to mention a few briefly, a variety of statistical and machine learning techniques can be used to effect a nonlinear, sometimes piecewise linear mapping between the inputs and the outputs, and I'll talk about that a little bit later. If you don't know the class membership, and I'll give an example of that towards the end, you use unsupervised or clustering methods in which you seek to align things that are more similar to each other with each other. There is also a much less common but also powerful technique, there's a talk tomorrow morning on semi-supervised, called semi-supervised, in which you know the class membership of only a subset of the data. And you want to use that knowledge even though you don't know the class membership for all of the data. A typical example in biomarkers might be Alzheimer's, where actually the only definite distinction of Alzheimer's or not is post-mortem. So often you don't know the class membership before the patients died. And so a typical so-called binary classifier problem of this type, in which you're seeking to take your metabolome data and determine the class membership, is accompanied by what's shown at the bottom in green and red, the confusion matrix, in which you have false positives, false negatives, true positives and true negatives, and various combinations and ratios of those are typically used to determine the quality of the analysis that you've got. However, although I'm not going to concentrate on the early phases of metabolome data gathering today, because that's a whole other talk, combining mass spectrometers and things like that, you cannot ignore the fact that if you don't get the experimental design right from the get-go and the data acquisition, you've almost got no chance, however fancy your number crunching after the event. It's too late. So you cannot divorce the big data analysis from the generation of the big data or even the small data in the first place. So in the very first edition of the Society Journal, we set down the steps that you typically do in a metabolome design experiment, which I've put there into 4 with red separate parts of it, really working out what you want to do, setting it up, getting the raw data, and the design of experiment I'll talk about in a second too. Then the data, of course, come raw, typically off a XCMS machine, or sometimes a one-dimensional NMR type machine, and there will be things that aren't right in them. The machine has drifted, the machine has done a dirty and just done it, or you've had a piece of rubbish in one of the test tubes, and there will be outliers, and really you need to identify them before you start any of the serious data analysis, and you may wish to normalize your data in various kinds of ways, including internal standards and stuff, but various kinds of normalization steps, in which you then come to where I had before, the analysis of canonical tabular data of your samples versus metabolites and other properties. And then, it says there, mentioned just already, typically you will start with some unsupervised analyses, including things like principal components, which you might use for detecting outliers and frankly not for much else. And then finally, you want to begin to turn the data into information and knowledge and visualization, although I don't have time to talk about it in any detail, I'll show a couple of things. A very, very important part of big data analysis, because if you want to persuade people that the data say whatever it is you claim, you need to be able to present it in a way that is intelligible and not a table of 10,000 by 3,000 figures in the form of a spreadsheet. However, danger lurks everywhere. Dragons lie almost wherever you look. And after one of the society meetings, Dave Broadhurst and I wrote this thing that tried to summarize some of the things that can go wrong if you just get your eye off the ball for a couple of seconds. One of the trickiest is so-called bias. In other words, where there is a covariation in something you're not interested in that goes with the thing that you are interested in. So in one rather notorious proteome experiment, all the cases were done in a mass spec on one day and all the controls on the next day, and oddly enough they determined the mass spectrometer drift rather than the difference between the case and controls. In another version, all the cases were men with heart failure taking pills, and the women were and the controls were women who weren't, and the bias was that you were looking actually at the women, not whether they got heart failure or not. So, and bias is probably the most difficult to control for. It really is very difficult because you don't always know what is covariant with the thing you're actually interested in. Also, less of a problem for metabolomics than for some of the other omicses, the transcriptome and proteome, inadequate sample size. You think you've got an interesting thing with a p-value that you regard as respectable, but actually the literature summarized regularly by John Ioannidis shows that this is very rarely done, and his famous paper, Why Most Published Research Findings Are False, is well worth a read. In addition, if you're trying to find differences between something and something else, and you've got 1,000 possible metabolites to test it with, you have to do, in the worst case, so-called Bonferroni correction, because you're essentially testing, if you've got 1,000 different metabolites, 1,000 different hypotheses. So if you think that a p-value of 0.05 is okay for one, you have to stick another 3 noughts in front of that to get to the same thing when you're doing multiple hypothesis testing. This is sometimes known as data dredging in the big data analysis literature, and it's better not done. And in addition, there are a slew of numerical methods, many of which are not necessarily that easy to understand. 2 talks following, and a talk a bit about making available the methods you did and some of this sort of stuff. But very commonly, you will find that people are producing models that are not adequately validated or cross-validated, or they use, for instance, a part linear method for disjoint data, or things that are very non-linear that have no chance of fitting But of course they will make a fit, but it doesn't make any kind of sense to use those sorts of approaches. And so those are the things we listed in that paper. The one we didn't list, because we kind of assumed that people would have done it, is the ones who don't actually use any stats at all. And that's even worse. And as I refereed 200 abstracts for this meeting, I barely saw one that mentioned any statistics, so better to try and note that one should try and incorporate them. Just one thing, pointing out that there are well-established ways by now of correcting things like mass spectral drift, but they require, to be done well, what people might think of as unreasonable fractions of the analyses to be of standards. In this case, it was 1-to-1. But when you do that, you get decent data, and it's much better to have half the number of things analysed properly than twice the number of things not analysed properly at all. So now again, assuming that we have in fact done all of those pre-processing things, and we've got ourselves a nice set of columns with at least names that are either real metabolites because you've identified them, or reproducible spectrally identified objects whose chemical identity you don't know, we then can move towards again our canonical form of objects against variables. And a very key point in big data analyses, you may have a spreadsheet of so many rows by so many columns, and the product of that is the number of cells, but the complexity of the data is not simply related to the number of cells. The complexity of a data set scales linearly with the number of objects, but exponentially with the number of variables. And metabolome data, of course, are replete with very large numbers of variables, normally much larger than the number of objects. Circumstances under which classical Neyman-Pearson statistics fail and don't work for reasons that very likely Jeremy will explain. So if you look in general at the relationship between the world of ideas, knowledge and stuff like that, and the world of data, or if you like big data down at the bottom, classically biologists are told they have to go down this right-hand arc, starting with a hypothesis or an idea and seeing if the data are consistent with it. And data-driven scientists or data scientists prefer to go in the other direction, in which they start with the data and find the hypothesis that best fits the data. And of course this is a cycle, and computers are involved for both purposes. The reason I've put that up is because it allows me to explain very simply that exactly in a similar form is the relationship between the otherwise completely different things called statistics and machine learning, even though both reflect a relationship between ideas and data. And again, statistics starts with a hypothesis and tests the goodness of fit of the data to that hypothesis. So again, it starts with a hypothesis. By contrast, machine learning and cognate methods starts with the data and finds the hypothesis that best fits the data. Those are two completely opposite philosophical strategies for dealing with big data, and you can probably guess the way I speak that I prefer the left-hand version of that, and again, you need computers to help you out. But statistics, I would say, when done nicely, can give you nice, easy to understand relationships, and here's an example taken from a classical biomarker metabolomic study, in which you can see very clearly, plotted out here as the p-value against the area under the rock curve against the p-value, a couple of very nice biomarkers that were properly validated. And so statistics has its place, even though I'm more a fan personally of machine learning itself, because the whole point of omics science is not to start with a hypothesis, to let the data tell you what is the best explanation of the biology in question. However, one shouldn't just fixate on the metabolomics, even though we're here at the Metabolomic Society, because in many cases we have other kinds of data that are relevant to the question at hand. And again, in 20 minutes I can't tell you all about it, but here's an example in which we use metabolomic and fluxomic data to relate to transcriptomic data and find that you can indeed map one onto the other in quite a nice way. In other words, with the metabolic network plus transcriptome data, you can predict all the fluxes. And as folk know, we and others have been involved in the reconstruction of various metabolic networks, including humans. That's the paper from a couple of years ago of the human one, and you can get it freely available and so forth. And to note that fully 1/3 of the reactions are transport reactions. That's quite interesting and important. And especially so, and I'm giving a talk about this tomorrow afternoon, understanding how the metabolites move around the cells in which one is interested requires these transporters. And we've had quite a story going now to the effect that the reason these transporters are used by drugs is because they're normally there for metabolites, and therefore the drug should look like metabolites. And so recently we published in the Society Journal an analysis of the relationship between a large fraction of the reconstructed human metabolic network, the metabolites therein, and a nice, freely available from Drug Bank, thank you David, file of all the marketed drugs, therefore drugs you assume that work, and we ask the question, are they in fact similar to each other or not? So, simple idea in cheminformatics, which is a sort of subset of big data analysis for small molecules, which I've not really got time to talk about at any level. There are ways of encoding chemical structures that end them up as a string of ones and noughts, and you can then compare those strings of ones and noughts with each other, such that the so-called Jacquard similarity, often in this field called Tanimoto similarity, is essentially the number of ones that or noughts that are the same in two things versus the total length of the string, the intersection over the union, if you like to speak about it in set theory. And you can then look one to the other, and you can find via what looks like and is a rather standard heat map, more commonly seen in transcriptomics, the clustering of the metabolites, the endogenous metabolites, and the marketed drugs, and you can see a fair bit of red and orange telling you that a lot of them are actually quite similar to at least one metabolite. And when you look through the data in full, using a whole series of encodings, we'll concentrate on the one called MAX, which essentially is a string of 166 ones and noughts that ask you whether such and such a group, OH or CH3 or whatever, is present or not. And you go from right to left, you can see that for 90% of marketed drugs, there is at least one normal metabolite that is similar to the drug that's your questioning drug. And so, again, going back again. So there is about 1,300 drugs and 1,100 metabolites, so over 1 million comparisons, so reasonably big data, but summarised pretty quickly, and it didn't take very long to do at all. And then you can interrogate it and say, there are all the metabolites, which are, and I will now interrogate it with a particular drug called metformin, which of the metabolites is the most like it, and it shows you there. Again, we're now moving again into the visualization, and fairly obviously you can decide whether or not that knowledge is useful, and in fact the metformin is transported into cells using a transporter called the organic cation transporter. That is, in its day job, the creatine and creatinine transporter. So the analysis tells you something that you think is useful. And then more recently, having recognized, and hence the rule of 0.5, at least one metabolite has a Tanimoto above 0.5. More recently, looking at the reasons, what are the substructural elements of the metabolomics data, those 1 million comparisons plus that we did, looking again at the various encodings, you can see that they give you very different likelihoods of having such and such a Tanimoto similarity, and it turns out that it's dependent almost entirely on the fraction of the encodings that have a 1 in them, various as opposed to having noughts, and that kind of explains why the encodings give such very different data. But again, it's quite quick to do. What we use, just a little puff at the end for it because we like it and therefore we're happy to sort of promote it. It's also free, which is a very big advantage, and shareable and open, and therefore everyone can use it, is a system called KINE, K-I-N, like that. And it's essentially a visual environment for constructing computational things. So if I take a typical screenshot of KINE itself, in the bottom left, you'll see all the sort of things I can pull in by drag and drop into the main part of the screen that do things, an elementary app, like read an Excel sheet, do a PLS in this particular example, or a genetic program, or a random forest, which are the three in this workflow, and separate the thing into training and test sets. So once someone reasonably geeky has constructed this workflow, people like me, all they have to do is change the file name, so it's very nice and easy. And all those heat maps and pretty things were generated using KNIME. So it's once you've got it up and going, it's really a convenient thing to use. And that's a close-up of the workflow itself. And if you go into the one called genetic program, which is down at the bottom called GP metanode in the grey, you will see if you click on it that you can tell it all the things you would expect in a machine learning thing like this, the size of the population and so forth. And that was written by my clever collaborator Steve O'Hagan, but once we've got it forever, and that's the attraction of this kind of thing. And there's a Python script hidden inside the node that does all the work, so it really is the same as a regular program, but it doesn't require me to write any code or know or care about a command line, because I don't think big data are going to make it into our community if it relies on command line users only. And it gives you a whole list of stuff there. And here's the results from a particular data set using two objective functions, and you can see that it learns to fit it quite well, both on the regression version of the ROC curve that I don't have time to explain, and the predicted versus the real versus the actual for a big data set taken in this case from Kell. And with that, I think I've used up my 20 minutes fairly exactly, and I thank you for your kind attention.
Many thanks for that, Doug. Do we have any questions in the audience to start with? I do. I've got one question for you. You've mentioned the importance of sample size and statistical significance. And as an early career member, obviously big data usually means big cost. And to get associated statistical significance, you need to run a lot of samples. Do you have any advice or hints or tricks that younger researchers can use to try and pull in bigger data size, bigger statistical significance?
So I'd say, and it was mentioned, but I forgot on the slide, but I forgot to actually say it. Fewer things reproduced properly is better than lots of things not reproduced at all for the same number of experimental objects. There are things that will tell you if you let your false discovery rate be 0.05 or whatever for standard name and Pearson statistics, then, and if the p-value is whatever it is, then you need X number of samples. And I would say that is completely the absolute minimum. And because people follow these things, that's why they keep finding out things that aren't true. And there is no real substitute for saving up your money to do fewer papers well, rather than just belting out stuff that actually has no significance. So it's not necessarily the message people want, but it is the truth.
It's a cool fact about all.